Welcome to this lecture on the neutral theory of evolution. When we think about evolution, we usually picture the fierce struggle for survival, where only the fittest pass on their genes. But what if I told you that the vast majority of changes in our DNA have absolutely nothing to do with survival? Today, we are going to explore a groundbreaking idea that completely reshaped modern biology. We will discover how pure random chance, rather than natural selection, drives most of the genetic changes hidden deep within our cells. Let us start by imagining a massive, ongoing lottery, where the prize is simply survival. In the traditional view of evolution, every genetic change is like a lottery ticket that either wins you a huge advantage or causes you to lose entirely. However, what if most of the tickets you draw do not change your chances of winning or losing at all? They might just change the color of the paper your ticket is printed on, which does not matter to the lottery machine. This is the core idea behind the neutral theory. It tells us that not every change in our DNA is a matter of life and death. Most changes are just harmless, invisible variations that accumulate over time by pure chance. Now that we have an intuitive understanding, let us look at the formal definition. The neutral theory of molecular evolution was first proposed by the brilliant biologist Motoo Kimura in the late 1960s. It formally states that the vast majority of evolutionary changes at the molecular level, meaning changes in DNA and proteins, are not caused by natural selection. Instead, these changes are caused by the random genetic drift of mutant alleles that are selectively neutral. An allele is considered selectively neutral if it does not affect an organism's ability to survive and reproduce. Its evolutionary fitness remains exactly the same. It is crucial to understand that this theory does not deny natural selection. It simply argues that selection only acts on a tiny fraction of all genetic changes, while the rest are just drifting silently. Building on Kimura's definition, you might be wondering why we care about changes that do not actually do anything. The neutral theory is incredibly important because it serves as the ultimate null hypothesis for evolutionary biologists. If we want to prove that a specific trait evolved because it was beneficial through natural selection, we first have to prove that it did not just happen by random chance through neutral evolution. Furthermore, this theory gave birth to the concept of the molecular clock. Because neutral mutations accumulate at a steady, predictable rate, we can literally count the genetic differences between a human and a chimpanzee to calculate when our evolutionary paths split. Without this theory, our understanding of evolutionary timelines would be completely broken. Knowing how important this steady accumulation of mutations is, let us look at the mathematics behind it. The most profound mathematical takeaway of the neutral theory is the formula for the rate of substitution. Conceptually, this formula tells us how fast new neutral mutations appear and successfully take over an entire population. The formula is beautifully simple. K equals mu. Here, K represents the rate of substitution, which is the overall rate at which neutral genetic differences accumulate over time. The symbol mu represents the mutation rate, which is the probability that any single gene copy will mutate in one generation. Notice what is missing from this equation: population size. Physically, this means that whether a population is large or small, the rate at which neutral evolution happens depends entirely on the mutation rate. If the mutation rate increases, the substitution rate increases exactly by the same amount. Before we prove why this simple formula works, let us see it in action with a concrete example. Imagine a population of 5,000 frogs living in a secluded pond. Scientists have determined that the neutral mutation rate for a specific gene in these frogs is 0.00002 mutations per gene per generation. We are asked to find the rate at which neutral substitutions will accumulate. We know our formula is K equals mu. We simply substitute the given mutation rate directly into our equation. You might notice we were given the population size of 5,000 frogs. However, this is extra information that we do not need to use because population size has absolutely no effect on the final substitution rate. The final answer is 0.00002 substitutions per generation. Physically, this confirms we can predict the pace of molecular evolution just by knowing how often copying errors happen, completely ignoring how many frogs are in the pond. Now that we have successfully used the formula, let us understand exactly where it comes from. We will derive this result in 3 logical steps. First, we need to figure out how many brand new mutations enter a population in a single generation. In a diploid population, every individual carries 2 copies of each gene. So if the total number of individuals is n, there are 2 times n total gene copies available to mutate. We multiply this total number of gene copies by the mutation rate, mu, which is the chance of a single copy mutating. This gives us the expression 2 times n times mu. This represents the total number of brand new mutations appearing in the entire population every generation. As population size increases, this number goes up because there are more individuals experiencing genetic copying errors. Moving forward with our derivation, we now have a pool of new mutations. But what are the chances that one specific new mutation will eventually spread to 100% of the population? This process is called fixation. Because the mutation is completely neutral, it gives no survival advantage. This means its fate is decided entirely by a random draw. Out of the 2 times n total gene copies in the population, only 1 is this brand new mutation. Therefore, the probability that this specific mutation will be the lucky one to eventually replace all others is simply 1 divided by the total number of copies, which is 1 over 2n. Notice the intuition here. As the population gets larger, the probability of any single mutation taking over gets much smaller because it has to compete with many more existing gene copies. This brings us to the final step of our proof, where we combine our 2 concepts. To find the overall rate of substitution, K, we multiply the number of new mutations created each generation by the probability that any one of them will actually fix. We substitute our expressions, 2n times mu multiplied by 1 over 2n. When we multiply these together, the mathematical magic happens. The 2n terms completely cancel out. This leaves us with the beautiful and simple result, K equals mu. The math perfectly proves our earlier claim. The high number of mutations generated in a large population is perfectly balanced by the low probability of any single mutation fixing. This is why the evolutionary clock ticks at the same steady speed regardless of population size. To truly cement this concept, let us look at a second example, comparing 2 completely different populations of fruit flies. We want to see exactly how population size interacts with mutation rates. Population A is a small, isolated laboratory colony containing exactly 100 flies. The neutral mutation rate is 10 to the power of -8. We will calculate the substitution rate, Ka, by multiplying the total new mutations by the probability of fixation. We plug in 100 for n and 10 to the -8 for mu. Because this is a very small population, it produces very few new mutations. However, each mutation has a relatively high chance of taking over because there are fewer competing alleles. The 100 terms cancel out, giving a final answer of 10 to the negative 8. Now, let us perform the exact same calculation for population B, which is a massive wild population of 1 million flies, experiencing the exact same mutation rate. We set up our equation, plugging in 1 million for n. This massive population produces a huge number of new mutations every single generation, which might make you think it evolves faster. However, the chance of any single mutation taking over is incredibly tiny because it is diluted in a sea of millions of other alleles. The high number of mutations is fighting against a very low probability of success. Just like before, the 1 million terms cancel out completely. The final answer shows that Ka equals Kb, which is 10 to the negative 8. Both populations accumulate neutral substitutions at the exact same rate, beautifully illustrating the core principle of the neutral theory in action. Having proven the mathematics, let us visualize this random process. Look at this graph showing random genetic drift over time. The horizontal axis represents time passing over many generations, while the vertical axis shows the allele frequency, ranging from 0 to 1. Each jagged line represents a different neutral mutation that has appeared in the population. Notice how they do not move in straight lines. They fluctuate wildly up and down. This is the random draw in action. Most of these lines eventually hit the bottom axis at 0. This means the mutation was lost from the population entirely. However, if you follow the bold line, you will see it randomly drifts upward until it hits 1.0 at the top right. This is fixation. The mutation has successfully spread to every individual in the population, becoming a permanent substitution. We have covered a lot of ground today, so let us summarize the most important ideas. First, the neutral theory fundamentally reshaped biology by showing that the vast majority of genetic changes are driven by random chance, not natural selection. These neutral mutations are harmless and do not affect an organism's fitness. Second, we proved mathematically that the rate of neutral evolution is exactly equal to the mutation rate and is completely independent of population size. Finally, remember that this steady, predictable ticking of neutral mutations acts as a molecular clock, giving scientists a powerful tool to trace the history of life on Earth. While natural selection shapes the traits we can see, random neutral drift shapes the vast majority of the DNA we cannot see. You now have a solid understanding of the neutral theory of evolution.